Hello, everyone, and welcome to another episode of the Modern Biopharma Podcast. Today, we have on Jake Adams from CRB. How are you doing today, Jake?
Wonderful. Thanks, Jesse.
Excellent. So I got connected to Jake through a mutual connection, and we chatted back and forth a little bit. He does some speaking on a number of issues. He's got really great history here in the pharma space, working at CRB. And one of the things that he has sort of spent some time talking about, thinking about, is the future of different ATMPs. And we have this sort of broader space where people talk about them, and certain ones get a lot more airtime, and they're a little bit more sexy in the space, as you may see. And he's got some thoughts on that. So we wanted to talk about that. And yeah, so that's what we're going to do today. This is going to be a discussion. It's going to be a little different than our normal one where I'm talking to a specific endpoint. I wanted to have just a wider discussion about these ATMPs and his thoughts and where they're headed. So that's what we're going to do. All right, cool. So Jake, before we get started, could you just tell everybody a little bit about yourself, where you're from, what you do, what makes Jake Adams Jake Adams?
Oh, yeah. Well, my good looks, first of all, which really won't transfer to the podcast format, which, we'll have to, look me up and really enjoy it. But no, I'm Jake Adams. I'm a Colorado native. I've worked with CRB for 10 years now, and really focused a lot on RNA therapies and kind of my journey there. I'm a Colorado native. I went to Colorado State, and I've been working here with CRB in Colorado just about the whole time. And so spent a lot of time in the RNA therapies world and really have watched that therapeutic platform kind of grow. And as we kind of have, as an industry, pivoted into this second wave of ATMPs away, or on top of monoclonal antibodies, it's been interesting to watch how some of that is sort of developed. And so I really took an interest in oligonucleotides, and of course had known about mRNA, but it wasn't really until COVID that hit the spotlight, and then did so in about as big a way as you
did it ever,
can.
right?
And so exactly. And so some of the powerful stories coming out with especially oligonucleotides and Batten's disease with Nielsen, which we can talk a little about, really caught my interest, right? And I think, like so many people in the industry, when one of these stories hits home, or you really hear the patient side of things, it's so inspiring. And so I really started to make a career shift towards learning more about these therapies and starting to drive CRB as a company kind of towards progressing this stuff. And obviously, many of us have been positively impacted now by mRNA. And I think that's just such an awesome story. And it was a real case of the power of the industry and all the smart people in it being realized and for a greater good. And so continues to just kind of build on that excitement.
All right. So I gotta ask a quick question before we really dive into the actual discussion about these different therapeutics and how they fit and all that. But we have a lot of people that listen to the podcast that are on the front end of their career, right? They're sort of coming in. And, when you came in, I looked you up on LinkedIn, and you had, this was not the first big project you worked on, right? You were not on an RNA project. You were on a huge, one of the bigger projects in the U.S., this giant blood fractionation site down in Georgia. And then you've sort of made the switch. And when some of these younger people come into the industry, first off, they have no idea. Like, it's like pharma, right?
Yeah. Oh, yeah.
But that's it. It's pharma, right? You're not thinking, I really would like to work on oligonucleotides, right? That's not how you do this. However, when you do come in, you sort of get into something, right? You get put on a big project, or you get put on it with a certain company that does a certain kind of product, and maybe you want to start to move towards something else. Can you just talk a little bit about how you did that? I mean, you're in a consulting firm, so maybe there's more opportunities there because you guys touch on everything. But how did you make that switch? Because you were pretty young. You're still pretty young. You're quite a bit younger than me. So, yeah, exactly. Those good looks, yeah.
Yeah, it's a great question, and it's something I talk a lot about with people I mentor at CRB. And there's no one right way to do it. But for me, working in plasma fractionation, one of the great parts about that project was the owner brought in lots of patients to the job site when it was being built to help kind of tell their story about how the drugs being made there really impacted them. And so I think that piece got me thinking more about additional new developments and new things, and hearing people who, a lot of what comes out of that plasma frac are immune drugs, immunoglobulin, IG drugs. And the part that hit home for me there was, hey, we're making this drug for people who maybe don't even have a diagnosed disease, and it's all the same treatment. And it actually, I had a sibling of a friend of mine who was taking some of those drugs. And so getting to kind of hear that personal story and having it connected there really clicked for me. And I was really interested in, call it blue water, right? Of saying, okay, monoclonal antibodies have been around a long time. Plasma fractionation has been around a long time. And there was a lot of expertise in the company that I absolutely, rode their back on learning. And I was interested in, okay, what's something kind of newer, right? Where I can develop expertise and it's not such a well-traveled trail. So for me, it started really easy. And I realized how much I didn't know, right? They don't teach this type of stuff in most engineering programs or architecture programs.
Definitely.
And so I started really simple. I started taking industry digests, right? And, there's a bunch of different daily email newsletters or weekly ones. And I just started taking those, and I would make it part of my own training and professional development to not just be learning how to be an engineer, but also learning what are these therapies? What do they do? And I'll be honest, I spent a lot of time on Wikipedia, and it's pretty good to give you a high level read when you're flipping through your daily newsletter of, stuff going on in the industry, and it's, hey, new clinical trial success for this type of thing. And you say, now, what is that?
I have no idea what you're talking about. Yeah.
Yeah, exactly. I'm going to Google it and get into Wikipedia. And so I was able to just sort of get a broader knowledge and then seek out some opportunities, right? The old proverb, right? Luck is at the corner of preparation and opportunity. And so I really believe that. And so, especially in the consulting world, but also outside of that, just having that base level of knowledge of, hey, I've been reading this stuff. I read the Wikipedia about it. You'd kind of be surprised how quickly, even at a high level, even for people who don't have a science background, you can get to sort of see some of these different things and find a little niche there. So it's not something.
So I'm gonna just interject here again, because I'm at a time in my life where I'm thinking about career, not for myself, but for my kids, right? So my kids are a lot old. How old's your kid?
8 months.
8 months.
8-month-old daughter.
That was a long time ago. So, and I've had 5 through that age. So, but my oldest is 18, getting ready to go off to college. And I've probably said this on multiple podcasts now, so it's probably old news. But, and you think about what you're going to go off and do. And I think about our space that we both operate in. And I'm like, if you go to college I don't care what degree it is. I don't care how specific it is to our industry. You barely scratch the surface of what's out there, right?
Oh yeah.
You don't touch it. And when you talk about that's, you're looking this stuff up on Wikipedia, and the number of people with deep knowledge on any of these things is quite small. It's really small, right? Because you just don't get there until you have broad knowledge, and then you focus it in on the one thing. And you can't do that without many, many, many years of school or many, many years of research. And then it's up to people like you to then digest that, right? And then feed it back to the broader population so that we can try to understand it and see how it's going to work. So I really appreciate just you sharing that, because I think for anybody that's young in the industry, like you're going into a meeting, right? And somebody like you now that sort of knows more, you're dropping these acronyms. You're like, oh yeah, this and this and this. Yeah. And they're sitting there just going, oh, I don't have any idea what he's saying, but I'm supposed to act like I do.
Foreign language. Yeah, absolutely. Well, and I think it's that. It certainly is daunting, from the outside of, wow, there's so much to learn here. But, you know, I recommend a lot of people this way of just start with something easy. Start with the industry news. Start to learn the names of the companies who are developing things. What are the buzzwords? What are the 3-letter acronyms that, you know, you've got to go figure out and write them down. Sit in a meeting when you're sitting there and having it all wash over you. Write down the 3-letter acronyms that are getting thrown around that you don't know, and go look those up. Go spend some time figuring out what those are. And I think it surprises people how quickly you can move through and start to get that basic parlance almost, the jargon of, okay, this is what this is. And you know, it's not going to be overnight, but it's not particularly difficult. You don't need to be, you know, subscribing to peer-reviewed journals for $400 a year.
Man, those are tough to read too. So those are,
yeah, even for me, right?
Yeah.
And so you don't have to be there to get a deeper understanding of kind of how this stuff works. So.
All right, cool. Well, hey, thanks for that. I appreciate that. It was a little rabbit trail that I just wanted to cover with you. So
absolutely.
What we want to talk about today, more broadly speaking, is this idea of ATMPs. I'm just gonna. It's Advanced Therapeutic Medicinal Products, correct?
Nailed it.
All right, got it. Bam. So people use across the industry, they throw terms around like this, and I think like we just discussed, you know, I think most people kind of know what it is, but do they really know? And do they really know the breadth of what that encompasses, right? That can encompass a lot of different things. So again, I hear ATMPs, cell and gene therapy. Boom. That's what I hear. Like, you know, boom. That's what I hear. And that's not necessarily, that's not the full, the full scope of ATMPs, but those are the two that get put together. And so what we want to talk about is sort of what's out there for ATMPs, and maybe what's getting the most airtime versus maybe what actually has some really good future that they're getting airtime too. It's just not as big, and maybe not quite the same investment too. So like just as a couple little things, in 2020, there was $20 billion invested into cell and gene therapy, which I believe was like a double what had ever been invested before.
Oh, yeah.
And then in 2021, $23 billion, which, you know, so that $20 billion broke all records. $23 billion broke it again. And I believe we're on track to do it again in 2022. And in 2022, there's actually a lot of, I believe, significant clinical trials that are sort of hitting, you know, big milestones. And so we're going to maybe finally start to see some actual usable therapies coming out of this space. Because as a broader sort of stroke on this space. So 23 billion invested in 2021. That's a huge number. That's a huge dollar figure for cell and gene therapy. And as of June 2021, there were 2 gene therapy products approved by the FDA. So 2 in total. I got this listed in front of me too. I think there's 21 or 22 total cell and gene therapy. So 22 total, right, in that whole space. And then when you include the other biologics that are approved, is 318. So biologics as a larger scope that all these sort of would fall under as well. That's a much larger scope of products that are out there. So it's tiny. However, you work for CRB, so you see this. How many facilities, how many companies are building cell and gene therapy manufacturing spaces right now?
That's right.
Like, is there any major pharma company that's not doing that right now?
No, you'd be crazy not to, right?
Yeah.
And I think a lot of what you're talking about really emphasizes the point that across the ATMP space, right, we're at the bottom, maybe starting to see the slope on this new wave. And as we go on over the next 5 years, the number of therapies approved in all of these different kind of ATMP modalities is going to skyrocket, right?
Right.
We really are understanding some of the science there. And yeah, there's just too much in the pipeline and too much investment and too much good to be done to kind of ignore it. So yeah, it's an exciting time to be in the industry from the perspective of we are about to open up this second or maybe even third wave of biologics, and it's going to change the healthcare landscape, certainly.
Right. Yeah. I sort of joke about it. Again, I'm a water guy, so I'm not really the science guy of the medicines, right? But I joke about how it feels Star Trek-y, some of the stuff. And I don't work in like the device space at all. But even in the device side, there's sort of contemporaries over there that are doing some crazy things too.
Yes.
Right?
Yeah. Outside of just true it's healthcare all over.
Some of the regenerative medicine things that are coming out, it's unreal. So if we could we just talk? So again, we say cell and gene therapy as a large cover. In reality, that's actually not. It's just a part of overall the ATMP landscape, and it's such a small part of the overall, even what's out there right now. So could we talk about some of the other ones that we talked right away? Everybody's heard of mRNA now. So these RNA drugs versus cell and gene therapy as more DNA drugs. Am I saying that right?
That's right. Yeah, that would be the right way to think about it.
So could we just talk about that, just those two? What's the difference in, like, say, an RNA drug versus a DNA drug?
Yeah, perfect. So you're going to have to bear with me on a little bit of biology 101, which might be taking you back to some old college courses. But remember that one of the most important pathways in your cells of how cells interact is protein creation, right? So your DNA, it's got a gene, and that gene, that sequence of base pairs in your DNA codes for a protein. And proteins, right, it's not just muscle, they do lots of stuff. They're really a very diverse set of molecules in the body, and they can be associated with communication. Enzymes are proteins. They're building blocks to cells, and so they're incredibly important. And what happens is DNA, a coding section of the DNA, is transcribed into messenger RNA, which leaves the nucleus, that core, the red dot in the middle of the cell where the DNA is housed, leaves the nucleus and is transcribed into a protein. And then that protein goes and does something in the cell. It helps transport things around. It helps communicate with other cells, whatever it does. And so our ever deepening level of understanding of disease and the way it's formed is actually starting to uncover a lot of diseases that at their root have to do with too much protein, too little protein, incorrect protein or non-functional protein. And so when we talk about cell and gene therapy and we talk about disease as a whole, right, we're starting to understand and be able to say, let's just take Alzheimer's, for example, even though this is a pretty complicated one, right? Alzheimer's has to do with a genetic mutation that's going on and your genes are causing some issue, or we're finding out that diseases have to do with genetic mutations, cancers, Duchenne's muscular dystrophy, stuff like that.
Yeah.
And so cell and gene therapy, DNA therapies are going all the way to the source. They are going to correct the gene that is wrong. And therefore, when it gets transcribed into messenger RNA, that's correct, and the right protein is being made. RNA therapies aren't going up to that DNA level. They're actually acting on typically the messenger RNA, but they're acting on something downstream of that DNA. But really the outcome, the therapeutic outcome is they're modulating proteins. They're fixing proteins, upregulating, downregulating. And so we can probably draw two pretty succinct buckets there of cell and gene therapy being DNA acting is curative. It's fixing the source code.
Right.
And RNA therapies are acting on the products of that source code.
Okay. Yeah. So I think that's what we hear a lot about, right? Sorry. I just interrupted you. Sorry about that.
Oh no, go ahead.
So the curative, and that's probably the thing we hear about the most, right? We hear about the cell and gene therapy being curative, which is again super interesting and amazing, and that's my Star Trek stuff.
Oh, it's super exciting. Yeah.
Right. And then again, so these RNA are it's more typical of a medicine, the way people think of a medicine. Maybe treating a symptom, but a little bit deeper than just treating the symptom. It's actually sort of treating a DNA symptom rather than inflammation, right? It's actually going in deeper and saying, hey, we don't want you to respond by using inflammation to do this, or you just go deeper and deeper and deeper, but you're sort of stopping before you actually mess with the gene in the body itself.
Exactly. Right. Okay. DNA therapies are going to be permanent. DNA is permanent in the cell. And so if you fix the DNA, that's permanent, that's curative. RNA, messenger RNA, is transient. It is made, makes proteins, and then is dissolved. Right. And that happens over and over. And so things that treat RNA are also going to be transient and continually dosed. And so they're not curative from that perspective.
Okay. So can we just talk to this really quick then? So there's a few different, I just want to focus a little bit more on this RNA for a second, because that's actually what's gotten the most airtime. I think it's funny. I said it at the beginning that the cell and gene is the sexy stuff, and it is. Right. It's super cool, and I think in our space that gets tons and tons of attention. I think in the broader, out in the world, just because of COVID, the RNA stuff has gotten a ton of attention, right? Even though that's not necessary, they're not necessarily related. Obviously, we're putting COVID facilities and new vaccine technology. It's coming out fast here, but that's actually really a pretty small spectrum of disease versus cell and gene is covering tons and tons, because there's so many rare diseases that I think those are going after. So if we could, just to talk to RNA, we got a few different types, and I'm cheating because you told me these ahead of time. So you have mRNA, you have we call them oligos. Us in the industry call them oligos. I don't, because I can't even say the word oligonucleotide. And then TPDs, and I'm just gonna look at this again, make sure I don't say it wrong. So targeted protein degradation. So those are our three. Mm-hmm. Are there more than that, or those? There's
a ton, right?
There's a ton.
Aptamers, okay. There's antibody drug conjugates. We see this all over, but yeah, really, with the RNA, we see a lot of oligonucleotides, and we see a lot of mRNA, and they're useful. They're useful two categories to talk about with this. So, starting with mRNA. So mRNA, and the principle behind the therapy is you are going to provide exogenous or outside mRNA into the cell that didn't come from the DNA. And your body doesn't know that. And so the mRNA gets in the cell and it starts reading that mRNA and making proteins. And so from the COVID-19 vaccine perspective, how that worked was, here's an mRNA that codes for part of the virus. And just one part, one of, call it 10, whatever it is. And as that mRNA gets into your cell, your cell starts making proteins that are really a part of the virus. Your immune system recognizes that protein's there and thinks there's an infection, generates an immune response, and there's the vaccine. So mRNA is making proteins where there wasn't a corresponding gene. Mm-hmm. When we talk about oligonucleotides, really what we're talking about is a couple different classes of RNA interference. And so what that's doing is inhibiting protein creation from existing mRNA. And so as genes are copied and it's making mRNA, it's saying, there's a problem with too much of this protein, or this protein is not functional and it's causing problems just in a buildup.
And Can you give me a quick example of what that would be for? So again, I just to go back, we know because of COVID, we've all gotten our own version of learning about that yep. it's sort of coding to make this protein, right? But when you're trying to interfere or stop the creation of proteins, what would that be for?
So far, a lot of rare diseases. We've seen it for spinal muscular atrophy, right, which is a problem with the development of the spinal cord and the nervous system. And so other diseases are rare, hATTR amyloidosis. But the perhaps easier to understand one is a relatively new oligo that has to do with high cholesterol. And so inherited high cholesterol there's a version of that. PCSK9 is the gene that they go after, and it has to do with creating enzymes to turn low density cholesterol into high density cholesterol. Right? If you've been to the doctor's office recently, right? They're like,
so we want fluffy commercial. Yeah. We want fluffy cholesterol, right?
Yeah, exactly. And so they're sort of looking at, okay, here's a problem in the cell, and they're using that oligonucleotide to downregulate enzymes that prevent that creation from bad cholesterol to good cholesterol. And so there's some examples there, but we really see it a lot still in the rare disease space.
Okay. All right. So that's good. I just want to make sure I'm properly understanding the difference there. So.
Yep. And I think one thing about mRNA that's important to remember is it's certainly gotten the spotlight as a vaccine, but the two big players, right, Moderna and BioNTech, did not start as vaccine companies. That was not the goal there. Right. And the concept of an mRNA vaccine has been around for 20, maybe even 30 years. And it was always just sort of, it wasn't necessary. We had traditional vaccines that are incredibly effective, safe, and work. And so there wasn't really a need. And I could talk at length and have about what's cool about mRNA vaccines and why they'll probably be the vaccines of the future. But the original goals were looking at oncology, cancer treatment, and how you could use mRNA drugs to help remove the way that cancer evades detection by your immune system. So your body innately has systems to destroy mutated cells, cancer cells.
Right. Yep.
And the cancers we hear about are the ones that are evading that immune system. And so looking at, are there ways that we can bring proteins into the cell that will help defeat that? And so mRNA is only just starting with COVID-19 vaccines. Really, the holy grail there are much broader, much more potent or currently undrugged diseases. And so with the influx of thinking and funding and manufacturing capacity, we're going to see mRNA drugs against cancer and genetic diseases as well start to enter the space in really cool ways too.
Okay, so is there you know what? Let's just touch the last one, which is the TPDs, before I ask any other questions there. So targeted protein degradation. So we have the first one is MRNA, which you're sending a message and saying, hey, DNA genes do this, right? Sending an email and getting it to do something. And then you have your next one was, I just had them in front of me, the oligos. And so that's going in and it's saying, whoa, stop, don't do that. Right?
Yeah.
And then you have your TPDs, they're doing a third thing.
Exactly. And so TPD is an interesting one because like we've talked about, you got DNA acting, RNA acting, and TPD is really protein acting. So the protein's already made. And one of the ways that diseases manifest is that there is a buildup of proteins that your body doesn't naturally digest for some reason, and they're non-functional. And you can imagine, for a simple way to put it, it starts to crowd cells and create problems and waste resources. And so targeted protein degradation is going to, once the protein's already made, it's going to flag that protein for destruction by the body. And that's done in a couple different ways. But again, it's kind of, we can think about it of, okay, there's this pathway of making proteins from DNA to mRNA to proteins. And we're starting to develop therapies that attack at all of those major steps. And so as we look at different diseases and actually what's manifesting that disease, we're able to hone in on the most effective way to specifically change what's going on and causing that disease. We're really targeting those therapies specifically for the disease.
Okay. So that's still considered an RNA modulating drug? That's in that category?
Yes. Yep. I consider it that way. I think, a sign of how new this all is to the industry is there's probably still some discussion of targeted protein degradation therapies being ATMPs. And really ATMP comes from some of the regulatory guidance and defines some of that, but it's really thinking cell and gene therapy. because that was a space that really needed regulations. And oligonucleotides and targeted protein degradation, because they're not such a sexy, to use your word, such a sexy new therapy, they've kind of snuck in under ATMPs, and still sort of defining all this and kind of bringing it all together is a much discussed topic, I think.
Okay. All right. So just to give it sort of the counterpoint, when we talk about cell and gene therapy, again, we're talking about curative versus non-curative on the RNA modulating side. One of the reasons we decided to do this was to talk about maybe the fact that the future for these RNA modulating therapies, it's significant, but it also may be doing a lot of the stuff. Like cell and gene therapy have some barriers that aren't necessarily, again, aren't obvious to people like me who aren't in the science of it all. So could we talk a little bit to what those are and why maybe those are, even though they're coming online and we're doing them, maybe they're not quite as close as we all hope that they would be or could be?
Right. So without getting too deep into science, right? Because there certainly is a scientific challenge that's easy to understand. And then I also want to talk about the social challenges, right? And so from a science perspective, when you're acting on the DNA, not only do you have to get the drug to the right cell in the body, you have to get it inside that cell's cytoplasm, or kind of the bulk volume of the cell. To get to the DNA and act on it, you also have to get into the nucleus. And so there's another wall, there's another boundary that you have to cross with the drug intact, with enough of the drug to be effective. And so there's some additional technical hurdles to get to the DNA, which is well protected by the cell. And so from a science perspective, there's that piece.
Okay.
From a social perspective, right, and talking about looking at, right, we're at the bottom of this wave, and what does the top of this wave look like? It looks like blockbuster therapies for common diseases. It's getting out of that rare disease space and really starting to realize broader patient population diseases. And it's not to say that orphan drugs and orphan diseases don't deserve their attention. They do, and these therapies will have impact there. But when we think about mRNA vaccines and how the COVID-19 vaccine went, right, we're probably sitting somewhere about 70-80% of the US adult population agreed to take an mRNA vaccine. And there was lots of uninformed counter messaging about what these vaccines did and what they would do. And so, there was a lack of trust there. And so as we start to think about RNA therapies, DNA therapies, and even protein therapies, right, these targeted protein degradation therapies, as we start to think about how that will be received by the populace, certainly in the orphan disease case, we have no other options, and they're typically, very bad quality of life or bad prognosis diseases. Of course, right? Everyone is going to be interested in whatever therapy they can get. The risk reward kind of
the more obvious, significant downside to an issue, the more likely somebody is going to accept a therapy that's looks, feels, appears more risky potentially, right? Yeah, because
exactly. And I think where cell and gene therapy will get inevitably is that we start to understand genetic mutations that predispose you to a disease, or are make you more likely to have a disease later in your life, and that we may develop a mastery of gene editing where we can fix that before it ever becomes a problem. High cholesterol, great. You have this PCSK9 issue, and we're going to edit that before you ever even have high cholesterol. And we will get there. And that will be so cool when we.
I have seen that movie. It just never ends. It never ends well in those movies, but I get it.
It doesn't. But I think in this case we will, and I think for that reason exactly, right? A lot of people look at that and go, "This is spooky," and are we now making brunettes blonde and this, that, or the other, or making everybody have a face like mine, whatever it is, and that's scary and has some ethical issues. itself, but, just focusing on disease, right? That's a pretty scary prospect. And I think it's something that is going to require a long time to build trust with patients and providers and manufacturers that that's a good idea. And so one could reason out what we saw with the social adoption of mRNA vaccines points to some challenges towards getting preventative, less serious diseases being cured, right? And editing your genome. And the science has some ways to go, but so do RNA therapies. And so we'll kind of get there. But as we start to look at RNA therapies, we've already started to see there's a drug that has been in Europe for maybe 6 months, a year, and just got approved in the US at the beginning of 2022 called Leqvio. And it's for high cholesterol. And it's an oligonucleotide. And it's going to be competing directly with statins, right? Which most people have heard of
like the Lipitor.
If you have high cholesterol, doctor puts you on a statin. It's an old small molecule API. There's a monoclonal antibody that is similar there. And now there's an oligonucleotide. So we're going to get to see this awesome competition, let's call it, of these three modalities: the old school small molecule, the first biologic wave monoclonal antibody, and dare I call it the second wave, this oligonucleotide, all trying to treat the same thing. And so both the monoclonal antibody and the oligonucleotide are sitting as second line therapies. So first you have to do a statin. If the statin doesn't work, then you can get prescribed this. But that may change. I suspect that will change as we start to see the success of oligonucleotides. And in that space, I suspect we'll start to see that shift towards first line indications. And to put it in high level, why I might think that is, one, statin is going to be a pill you take daily. And Leqvio, I think it's sitting at a twice year shot in the arm, right? And so, okay, it's a shot in the arm, but it's twice a year. You're not taking a pill every day. And while that alone, you might sit there and go, is that so transformative? Think about it this way. And hopefully you've been blessed with your parents, growing old, and or your grandparents before them. And you might remember that folks in their 90s and their 80s start to get on these pretty brutal regimens of, okay, I take a statin for my high cholesterol, and that gives me kidney issues. And so I'm on another drug to treat the side effects of that first drug. And actually that gives me this. And so I'm on a third drug. And so for one disease, you might be on 3 drugs to treat the disease, the side effects of the first drug, the side effects of the second drug. And that it leads to these huge pill boxes that you got to remember to take them all. And it's really sort of this not so great patient experience.
I remember my grandmother specifically. I think my mom was on it towards the end as well, but just seeing this giant, like the Monday, Tuesday, Wednesday,
exactly. And that's commonplace, right?
So with this, I'm just curious, and again, forgive the ignorance here, but like the closer we get to go to the sort of the source of some of these issues, it doesn't, so say we're not curing, so we're not going all the way to the editing DNA so that our body is no longer creating or contributing to that problem. But so we're going in and we're saying, whoa, enough of that protein, or hey, do this instead, right? So we're going in and would the overall side effects decrease with that sort of precision of medicine versus if you go towards your sort of chemistry side of the equation, like what we've done in the past? That's going in and it has a lot of sort of unintended recipients of activity. Like, so it's going in and we know that it helps with cholesterol, right? But it's super indiscriminate. And so other parts of your body are responding with all kinds of other things. So if we do this, you're going in and going a lot closer to the source, and it's not banging onto other things as it gets to that source. And so you end up with less sort of subsequent medications as a result.
That is exactly right. We're more targeted, right? Okay. We're saying this is the protein that's causing the problem. Let's act on only that protein. And so we end up with less side effects. And so even though they may not be curative, it may not be this holy grail, it may not be so sexy, RNA therapies that take that big step towards more targeted, closer to the source of the problem, end up with less side effects. And so we don't see, the future looks like we're not seeing these big waterfall of drug to treat the disease, drug to treat the first drug, drug to treat the second drug. We're really kind of honing in, and that's going to be transformative. That is going to make that health landscape so much better, right? And I think when we think of cell and gene therapy, it's so easy, especially for all the scientists out there, to just go, wow, that is so cool. What a cool, nifty, awesome way. And they're so proud of that. They should be. That is so, so cool. But I think we also need to remember that even for patients, right? And if we put patients first, thinking about how even these RNA therapies are maybe more acceptable to them, something easier to kind of sink their teeth into, that this is still going to be really transformative for patients. And as those RNA therapies get into higher patient population groups, they'll be quickly and easily adoptable and be a much better outcome for people. Even if they're just equally as effective, right? And I think we're going to see them be more effective in some cases. Even if they're equally as effective as the current treatments out there, that dosing, the side effect issues are going to be transformative.
And that's the way it is already with even more traditional medications. If you can take it once a month instead of every day, that's usually looked at as a better option, right? So,
absolutely.
Or people take about like the slow release capsules versus fast. So instead of every 4 to 6 hours, you take one a day, right? So there's no doubt that there's just inherent value in that difference. My exposure to this in my normal sort of just life and the way people think about this is when people fight things like this or get upset about it, they're just trying to come up with an excuse to be against something. But
yeah. Well, there's a whole nother podcast, Jesse.
So it's so funny is like I'm like, oh, I want to talk about so many different things right now. Like, and one rabbit trail I just want to ask about is in this world, in our COVID world and taking vaccines, one of the things I've heard a lot of people talk about is, why are we taking this experimental mRNA vaccine when we could be doing the tried and true monoclonal antibody like that's available, right? Like that's a treatment for it. And in a lot of respects, it's I don't know. There's a lot of they're not the same. They're not the same, but they're kind of on the same level of complexity. And there's a lot of things going on with both of those, right?
Yeah.
So when you hear that, being a guy that probably knows this stuff at a much deeper level than I do, what would you say to that? Would you say that's there's like a super false equivalency there? Like this is mAbs are really complicated and they're not super simple to just make them, make these. And yes, they've been around for a long time, but this is a whole new one because it's for this instead of for something else.
And maybe the better way to put this, right? Because really when we're talking mRNA vaccine versus monoclonal antibody treatment, monoclonal antibody, you've got to already have the disease.
Right. It's to treat a disease rather than to prevent.
Preventative vaccine or prophylactic. And so maybe a good example of putting this is, okay, there's, I'm sure, lots of people who got, their flu vaccine or MMR or, the standard regimen of vaccines, which are attenuated or inactivated viral vaccines. So if we just were to go, okay, equivalency vaccines, why would I take this experimental mRNA versus a more traditional style vaccine? I think the first answer is the mRNA vaccines are not all that new. Moderna especially had developed a lot of mRNA vaccines for chickamauga, H1N1 that got authorized for emergency use and so went into people. But I think it's almost the reality of the situation is almost the opposite of what people think. And the reason why it took so long to get from emergency use authorization of mRNA vaccines to approved is because the regulators were being super careful, right? They understood that just because there was an urgent need, we couldn't throw away all of the protections we have in place to protect patients. And that now, especially, the mRNA vaccines are probably going down in history as one of the greatest clinical trials ever done due to the sheer number of people that were enrolled in that clinical trial. We have more data about mRNA vaccines and how they're tolerated than just about anything we've ever done. And so it's a fair thing to say, this is new and I'm not sure I want to be a guinea pig. And I think the first thing is you're not. mRNA vaccines have been developed and been in use for a long time. Fortunately, those previous epidemics ended before it was really required to go through the whole approval.
I really didn't even have a chance to be all experimental around the world because they fizzled out.
And so, and now going forward from here to people who are still hesitant, it's hey, these were approved under one of the most robust sets of data ever. And now we understand the safety profile of these better than just about anything because billions of people have taken them. And so
how do the costs compare? How do the costs compare? Like, so the monoclonal antibodies is a treatment. So, if one person gets the MAB treatment, the cost for that is significantly higher than a vaccine, correct?
Yeah, much more difficult to produce. Monoclonal antibodies or traditional vaccines, you need a bioreactor. You are growing cells that express that biologic, harvesting that, purifying it, putting it in a vial ready for people's arms. mRNA is actually a much more simple process that starts from an engineered DNA template, typically a plasmid circular DNA, and is just copied. And so making tons of mRNA vaccines requires much less volume, much less steps. You're never growing cells. It's never part of a whole cell. It is just mRNA. It's easier to purify. It's a cleaner process. It's slightly less susceptible to infection or contamination. And so it's done at a smaller volume to get the same number of doses. The manufacturing spaces tend to be a little bit easier, more deployable. And so, the cost difference is just huge. And I think the other thing we're starting to see with mRNA vaccines is the platformed approach. Okay, here's one mRNA vaccine, and now, God forbid, knock on wood, COVID 2022 comes around, right? And COVID-19 is replaced. Well, the amount of changing and tweaking of that mRNA vaccine that's needed to modify it is going to be relatively minimal. And so there's an argument to regulators, which is being made and under evaluation, is that we already know the vaccine itself is safe, and we know that the mRNA in there is safe. And if we can just prove that the slightly tweaked sequence of mRNA is effective, then we've already got all this data to show it's safe. And so accelerating through that clinical trial, and hopefully mRNA vaccines get to a point where we can really be meeting the response or the need much quicker.
So we can get to a meaning. So again, I guess at this point, the idea is, all right, does the vaccine that we've all taken is it really effective anymore against these new strains? Everybody argues in a million different ways. I won't even touch that, so don't need to. But we got new strains. Let's all agree that it's less effective.
Sure. Right.
Okay. So certainly that. But with new strains, we can respond a lot faster. With a MRNA vaccine than maybe a typical, or is it the same, or how is that what you're saying?
That's exactly what I'm saying. Is even if you take away my argument of, hey, this could be a platform system and really accelerate things, the manufacturing capacity is there. The understanding of how to do this is there. And so as soon as we've sequenced the genome of the new virus, COVID 2022 as we're calling it here, we could make the associated mRNA and put it into use. And so the more rapid response to help prevent the spread prevents it from becoming this pandemic, keeping it in an epidemic, a localized infectious scenario. And so absolutely quicker to develop, easier to develop, cheaper to develop. The manufacturing facilities stay exactly the same. mRNA facility overnight could go from COVID-19 vaccine, COVID 2022 vaccine. And so the potential there is very good.
Yeah, I mean, that's a big deal because in our historical facilities, and I know this is the kind of thing that this is what makes CRB, CRB, right? Because you guys are really pushing that envelope and trying to figure out new and better ways to have a manufacturing facility that can transition from one thing to the next, or back and forth, or whatever, right?
The multimodal speed to market thing is,
right.
is key.
And that was unheard of 20 years ago, I think. Right? It was just big, like lots of stainless steel, go.
And huge bioreactors, and we're going to crank this out for a decade.
Right, and I've been to all those, or tons of those facilities, and seen them, and there's still plenty of things that we need that kind of facility.
Absolutely.
But with the ability to address so many different types of things with some of these new technologies, including RNA and DNA therapeutics, we can do so much more targeted that you have to have the ability to do multiple, because the more targeted, the less, I guess, the less volume.
Less indications.
Right. Yeah. The less volume you're going to be addressing of people, of arms or whatever. Right. So
absolutely. Yeah.
So that means that everything's going to get smaller and smaller and smaller. And so to address a wider market, you have to do more different things in order to get there.
That's right. These flexible facilities are certainly the landscape of the future.
Yeah, I mean, it's we've been going for a while here. I feel like we could just keep spiderwebbing off to talk all these different things. I have more questions, and
I hope a lot of your listeners hear this and have other thoughts on this, right? Because I'm putting an opinion out there, and it's not the only one, and I'm not the most informed, and so I think this is a discussion that should continue to be had in the industry. It's something that I want to raise the issue, because I think it's important that we don't overlook RNA therapies and the importance and the changes that those could bring about for the better.
So can I want to just this, I think we can just close up with this idea. And then, I think I want to do this again with you. You're super fun to talk to. So,
we'd love to.
What I'm curious about is, so I think we both operate more on the side of the building of a facility.
Sure.
We're not, correct me if I'm wrong, but you're not really driving on the front end of exploratory stuff, right?
No research and development.
Yeah.
No.
So you're not on the R&D side, neither am I. So we're really more on the receiving end of the R&D, right? Like it got this far, and now we're like, hey, this has been effective enough to build a facility for it. So that's really where we're at. So as you think about this, and you're going, hey, I really think that these things could be really good. Why aren't we talking more about these? At the end of the day, that's not actually where we exist. We don't actually exist in that sort of influencing space. Right. So where and when does that have to happen to make sure that we're not missing a boat? We're not just, pouring a ton of money into one thing because it's so amazing, or it could be so amazing. And when in the meantime, we could be doing more of a different sector of therapies that, maybe are a more appropriate stepping stone, right? So,
right.
It's like, we're trying to go from horse and buggy to, a spaceship, and like there's probably a nice gas powered car in the middle that would get us there, right? So
that's right, and I think you're exactly right. The other thing that's true about this is what we're talking about here, and the potential of RNA therapies is not really overlooked by, let's just call it big pharma at large, or the industry at large, and that RNA therapies exist in a lot of pipelines, and they're not talked about because they're not in the news, right? It's a less easy soundbite for your, morning national news broadcast to talk about.
Mm-hmm.
But I think on the R&D front, there's a ton of great progress being made to really open up the possibilities for these advanced therapies, and especially these RNA therapies. And so super exciting time. And I would love to do this again, and we can start to unpack some of the technical challenges that exist with some of the cool stuff I'm reading about, which if I'm reading about it, it's probably a year old or two. And so I think we're going to see a lot of movement here, because there are a lot of smart people.
All right, so I need to start reading more Wikipedia to catch up with you, since I've heard that's the path to success in this space.
Hey, it's nice and easy, nice and digestible, good pictures usually, which help.
I've done it plenty of times. I remember when I first started in some of this stuff, and I'd end up with somebody talking about a kind of valve or something, and I've not done the hands-on stuff, and I'm like, okay. So I would go and look it up and find pictures, and then you see diagrams that are like, okay, conceptually now I get it, and then they'd be like, oh, well, you just got to take it apart over here, and I'm like, I got nothing. I don't know what that means. So then you go and you find a YouTube video where a guy's taking it apart, and you're like, oh, that's what he's talking about, right?
Exactly.
So I've done a similar, far less technical version of the same kind of thing. So. Hey, well, Jake, this was awesome. I appreciate it. I've been taking a few notes here, and I got some ideas for another one of these sometime. But
looking forward to it.
Thanks for doing this. Maybe next time I'll come to Colorado and we can do one in person instead of from across the country. What do you think?
Yeah, we'll do it at the base of Vail Mountain. In between a couple runs.
All right, I need some new equipment for that, but I'm game.
Deal.
All right, thanks, Jake.
Thanks a lot.